Pandomics is a powerful tool specifically designed to empower pharma drug and target discovery pipelines. Pandomics combines omics, text, and chemistry databases in a single solution available online at pandomics.com. It implements a huge part of the comprehensive end-to-end AI-driven drug discovery pipeline carefully designed by the team of Insilico Medicine in the recent years. A number of papers in the top-tier journals are featuring Pandomics and its core algorithms. To apply for Pandomics, just press the Request Access button and fill out the form. We will get back to you shortly and provide your login information. Pandomics is in the alpha stage, which means that you can get a significant early user discount if you apply now. The first thing you see when logged into Pandomics is a comprehensive internal omics database. The current version of the database includes data for transcriptomics, including both microarrays and RNA-seq, proteomics, and methylomics. Integration of SNP data is under active development at the moment. The important advantage of Pandomics is a single interface that allows you to access the omics data from multiple sources in a standardized way. Users can upload their own data and combine it in a single analysis with the data from the internal database. Let me show you an example how Pandomics can be used for the analysis of a particular disease. All the samples for the particular disease or condition can be found in the database using the search interface. The additional search conditions such as sex and age of the donors or even the particular tissue type can be added. The sample interface allows to browse through the samples, sort them according to the particular features, and create groups for the subsequent analysis. To run the analysis, further referred to as experiment, the one or two groups should be selected. The experiments themselves are organized into projects that can be shared to other users. A number of additional settings related to the gene and pathway analysis algorithms are available through the interface. Each report page contains several tabs. The Gene Analysis tab provides an access to a basic type of analysis, the differential gene expression. The results are present as a so-called volcano plot, which reveals genes with statistically significant differential expression. These genes lay in the upper left for downregulated genes and upper right for upregulated genes, corners of this plot. For each individual gene, you can find the corresponding values of the fold change, describing the difference in the level of its expression in the case, group versus the control, as well as the p-values, describing the statistical significance of the found difference, in the table below, where the search is also available. The values in the table can also be sorted, giving the most up and down regulated genes. While pressing this button, one can see more details in the statistics. Moreover, for each gene, there is a list of potential drugs associated with it. Clicking on a drug, one can see more details on it, including chemical and pharmaceutical information, as well as the efficacies and side effects predicted by the AI-driven algorithms developed by Insilico Medicine team. On the Pathway tab, you can check the results of a more sophisticated type of analysis based on estimation of the up and down regulation of molecular pathways. The user can choose between several algorithms for pathway scoring, including iPanda, our proprietary one, which shows several advantages over more standard approaches. Using the search field, any particular pathway can be easily accessed. For each pathway, the details about the individual genes are available by clicking this button. The diagrams of pathways are colored according to expression levels of individual genes. Red corresponds to upregulated ones, while green to downregulated ones. This plot is interactive so by clicking on a node, you can see all the neighbor genes interacting with it. Also, in the bottom part of this page, one can see the heat map showing scores of all or the selected pathways for individual samples for which the current analysis was carried out. The Drug Scoring tab allows one to look for chemical compounds which induce alteration at the level of molecular pathways complementary to that in the condition of interest. Such compounds can thus counteract the malfunction and be potential drugs. The gene expression profiles for these chemicals are available for a number of cell lines, so you can select one which better resembles an appropriate tissue. Each entry in the Appear table with results is cross-linked with the PubChem database. One of the core features of Pandomics is the Target ID tool. It utilizes the information from multiple publicly available sources, including omics-based network methods, 3D structure availability, information on the known inhibitors and drugs, clinical trials, grants, patents, and scientific publications. Pandomics inherits the results of the text analysis tool called pharmacognitive.com, available online for free and designed by the team of Insilico Medicine. This tool allows to capture trends in a particular field of research and analyze the level of trust to a particular journal, organization, or even individual scientist. Pandomics combines this data with the output of the omics analysis, resulting in an easy-to-use ranked tables of genes based on their potential relevance to the particular disease. The list of compounds known to inhibit or other way around activate the particular protein can be easily found through the interface.